and using the 3D organoid culture system and RNA targeting RBPs. Go ahead, Gene. Thank you.
Great. Thank you, Beth, and the organizers for inviting me to present some of the work from my group. So I'm not going to talk about mice models or ASOs, siRNAs, or even muscle. So I hope you apologize. I'm going to talk about something different and give a high-level perspective of how we think that this is distinct. So before I do that, I'm going to disclose statements of financial interest, particularly to the Locana Bio, and also my current funding sources here at my lab at UC San Diego. Okay, so just to give you something a little different, just start from cells. There are a lot of proteins in cells known as RNA binding proteins that are encoded in the human genome. And these proteins bind to sequence-specific recognition motifs or structure within hundreds of thousands of transcripts. And so work in my lab has been focused on thinking about RNA binding proteins and understanding what they do. Normally to human biology, molecular biology, they affect processes like splicing, RNA localization, stability, and translation. And most RNA binding proteins have two components to them. One part recognizes the sequence or structure, and the other part, sort of effector domain, controls a level of processing here. So many steps you can alter, obviously, with RNA binding proteins. We've also been thinking about RNA binding proteins as drugs, and so I'll sort of mention our thoughts in that area. And more recently, we've been moving into thinking about RBPs as drug targets. So just again, more sort of background, RNA binding proteins bind regulatory elements, and they can be expressed in cell-type-specific manners. They can control molecular aspects of gene expression regulation. And over the years, we have been developing methods to identify, at nucleotide level resolution where RBPs bind. And some of this work has been, you can access at a company that we spun off from my group, Eclipse Bioinnovations. Some of the technologies there that could be helpful for others in the RNA therapeutics arena. And then we've been leading community efforts to really provide the largest maps in the world on where all these RBPs bind, where they go, and what they do in human cells. Now, through this sort of decade of work, we have came to the realization that RBPs are very interesting to study, but they can also be engineered to do things that we want, right? And sort of the first instantiation of this took a place in my group in 2016, where we were repurposing CRISPR-Cas9, which in our mind is a RNA binding protein, in this case guided by an RNA, but still an RNA binding protein. And we're able to show that when you fuse GFP to it, we were able to track RNAs in live cells. And then quickly following up this work, postdoc Ron Batra in my group led the effort to show that you can then fuse the RNA targeting CRISPR proteins to endonucleases, like the pin endonuclease, to degrade toxic RNA. And toxic RNA can be obviously, as you already heard, be found in the myotonic dystrophy type 1 or DM1 locus, right? ALS, for example, C9 ALS, and also in Huntington's disease. And so we showed this was an interesting new strategy to develop AAV gene therapy and deliver this in cells, and then you get a permanent depletion of toxic RNAs. Recently, Ron published this work with my group that showed that you can actually show in preclinical mice models all the reversals you have seen in Ramesh's beautiful talk using oligos, but we can see this with RNA targeting CRISPR approaches, including obviously reversing mice behavioral phenotypes seen in DM1. Over the last years or so, we've also been developing ways to modify RNA in a single nucleotide manner. Now this is sort of a broad stepping back. This just gives us one set of tools that can recognize RNA in a programmable manner. How do you create other activities from these RNA recognition domains? So the other set of work in my group focuses on basically taking all the human RNA binding proteins, doing very large-scale screens on thousands of these to identify domains that, when brought close or to a specific RNA, can do whatever you want to that RNA, be it splicing or stability or translation, and so that provides new functional modalities to targeting RNA. And when you combine these two, we wrote in a recent review, you have this RNA-guided or completely protein programmable systems to modulate and target RNA. And some of these concepts have been realized and pushed in this company from my group, Locana Bio, that's in San Diego, pursuing RBPs as drugs. And so today I want to talk to you about C9orf72. And so again, this is a great model for us to think about disease pathogenesis as well as therapeutics. And because this hexanucleotide repeat expansion is the most common genetic cause of ALS, right? And you know, there are many hallmarks of this disease. You get upon disease onset, it's a rapid degeneration of the upper and lower motor neurons, right? However, we also see that half of C9orf72 patients experience cognitive impairment. You know, many of these patients are also affected by frontotemporal dementia. And so we've been thinking a lot about two aspects here. How do you model the neuronal aspects of the disease? You know, unlike our studies in myotonia dystrophy, you can't just go grab, you know, where you can get muscle biopsy from patient, you cannot really go get living neurons from patients affected by C9 ALS, right? So how do you model this disease? And then what are ways to target this toxic RNA? And so just as a little bit more background, this is the C9orf72 gene found and others have found that this G4C2 repeat is in between exon 1A and 1B. So in the intronic region, this large expanded repeat can be seen and expressed as RNA both in the sense strand and the antisense strand. Antisense is basically, you know, expressing C4G2, but both sense and antisense RNA repeats form foci, and then this foci can sequester RNA binding proteins in living cells to lead to repeat RNA-mediated toxicity, but they can also leave the nucleus and be translated to form dipeptide repeats, which can also prove toxic to cells. And there are also studies that indicate that C9orf72 may have haploinsufficiency as a cause of disease. So there are many multiple mechanisms. In our minds, we were wanted to see which mechanisms are initially present in neurons, for example, from patients with this mutation, and then understand also can we develop ways to modulate this, right? Okay, so Kathy Morelli, an excellent postdoc in my lab, sought to first model this disease in neurons, and one of the most exciting directions in the stem cell arena has been to develop iPS lines into 3D organoids, and so Kathy utilized a protocol that was developed in Alison Motri's lab at UC San Diego, our next door neighbor, to push wild-type iPS lines into mature forebrain cortical organoids, and you can see it takes months and months to develop, you know, CTIP2 positive cells and that reflect the right neuronal connection. Now, what we also obviously want to do is use develop these iPS 3D organoids from patient fibroblasts from C9orf72 patients. So these are fibroblasts obtained from our collaborator John Rabbits from C9orf72 mutant individuals, and these were then reprogrammed into iPS lines in my lab, which then Kathy utilized her protocol that she's been adapting from the Motri work to then differentiate these into cortical organoids to model what we hope, you know, would be cortical dysfunction or neuronal misregulation. And so this is still very early work, so we can't really tell you too much about whether we have seen clear phenotypes yet at the cellular level. But what was very exciting though is we do see that the C9 cortical organoids and our control organoids differentiated readily, into these C22 positive cell types over months. So this is 2 months. By 4 months, they start developing mature synaptic markers, VGAT and neuroligin. And so that's again takes a bit of time to get the cells to this stage. And then at 6 months of age, they start also expressing markers, and you can see at the single cell level astrocytes within these 3D organoids, right? And so we do see that we can get mature 6-month-old cortical organoids from C9 and CVB patients. But what's exciting though is actually that even at 1 month of age, these organoids are starting to express G4C2 ribonucleic foci. And so the RNA foci show up very early on in these cells. And keep in mind, this sort of represent early, prenatal development, right? And so it's pretty exciting milestone for us to see that these organoids can actually display this G4C2 ribonucleic foci. And then when you look at later on, the dipeptide repeats start showing up at 4 months of age, right? So as the cells mature even more and the the 3D organoids continue to form connections, right? The neurons form connections between the individual cells. You start seeing these dipeptide repeats also appearing in these organoids. Now the other organoid that we've been very excited to try to make that Kathy and others in the lab has been pioneering is like making spinal organoids which contain CHAD-positive motor neuron markers and islet-1 markers. And so this is again still relatively new. It takes months and months to differentiate these to maturity. But I think we are hoping to use both of these systems to then test engineer RNA binding protein targeting approaches. And so at the last couple of minutes, I just wanted to sort of point out what we've been very busy doing. So Kathy and others in my group have been now taking the menu of RBP specificities that we extracted from our studies to then combine specific domains with, again, endonucleases that can chew up RNAs when brought close to RNA. And so the RNA targeting domains, we hope, right, would be able to recognize, for example, G4C2, or in this case, also the C4G2 repeats found in this C9 or 72 loci. And with the destruction of this foci, we have tools now then to then understand, is it the foci, the RNA foci driving, the disease in human systems, not mice, right? And can be, or is it the dipeptide repeats that appear later, at 4 months of maturity, for example. So I can't really describe too much details yet on our final modalities here, but just to keep in context, we published in Cell 2017 this RNA targeting Cas9 approach used to PIN for DM1, for ALS, for other repeat disorders, and then obviously there's been an evolution of other systems now, that us and other labs and Locana Bio has been developing, like Cas13D, and then the puff proteins that can also be programmed to bind RNA. And so today I'm going to just show you this very small one that we've been developing, which is what we call Z1 pin. We can't really disclose the domain yet, but it is so far the smallest programmable system that can bind and target G4C2, but also surprisingly C4G2 repeats. So just as a molecular sanity check, when Kathy expresses C4G2 or G4C2 repeats in cells, just to show that we can test the specificity of the system, right? You can see that in cases where we express AAV or AAV9 package Z1 RNA binding domain fused to the PIN endonucleases in the spinal organoids, we're able to destroy this C4G2 or G4C2 repeats in just with one RBP fusion, right? Which is pretty exciting. You think that maybe this just hits any GC-rich region. That's not true, because when we test with CUG repeat, which is found in obviously DM1, DM2, or CAG repeats found in Huntington's, we do not see a reduction right? The first dot plot there is basically just expressing the repeat alone with, not expressing the repeat, so it's blank, but we don't see any reduction of other GC-rich transcripts. And as I mentioned, these Z1 pins are transferred into AAV9 because AAV9 has great tropism for the neuronal cell types we care about. And so if we put these things together, we actually get beautiful biodistributions in our spinal and also cortical organoids. In green here, you can see that the Z1 pin, we also tagged it with HA, expresses quite well in these organoids, suggesting that the combination of the organoids and this gene therapy approach is a good system to study the progression of the disease and for therapeutics. And excitingly, we were able to show that we can destroy both sense and antisense repeat with what we think is the smallest version of RNA targeting protein-based therapeutics to date, which will be a one-time, hopefully, injection in patients. So very early, very odd and different, I know, from the other talks that we've seen, but hopefully this is an interesting direction. With that, I want to thank everyone for paying attention, and definitely Kathy Morelli from my lab that's led all this work, and my group at UC San Diego. And I'll stop here.